Okay, so this question is really just a short refresher on arterioles, and specifically on our understanding of vasoconstriction and vasodilation versus receptor types and neurotransmitters, because I know that was a bit of a hefty topic. We have a little more kind of hefty mathy topics coming up today as well, but I want to make sure that we got this receptor stuff straight. Okay, so we're looking here at vasoconstriction, thinking about what neurotransmitter is going to cause that, whether the amount matters, and whether our receptors make a difference. Okay, so really to answer this question, I'm going to walk us through drawing a quick flow chart of our two neurotransmitters that we're tracking and our two receptor types. Got another person joining us real quick. Great. Okay. So this is a flow chart of transmitters. Okay. You don't need to be read. Okay. Our neurotransmitters. Okay. So we have two neurotransmitters that we're thinking about. Both are associated with our sympathetic nervous system. So we don't really think about parasympathetics when we think about changes to arteriolar radius. We're just thinking about that sympathetic component of our autonomic nervous system. So we're thinking about norepinephrine. We're thinking about epinephrine. So the reason I pulled this chart on the bottom out again is we learned this stuff in words on the slides, but this is a handy dandy little reference that tells us what these two neurotransmitters are going to bind to. So we can see that norepinephrine is really binding to our alpha receptors. And remember that we're comparing here alpha receptors and beta 2 receptors are the ones that we're interested in for our blood vessels. Alpha receptors are kind of everywhere. Beta 2 receptors, we're specifically in this case going to see in our skeletal and cardiac muscle. So that differs a little from this chart because it's from a previous chapter and has some different focus, but we're going to draw in here our receptors that we're affecting. Okay, so norepinephrine, we're going to see affects our alpha receptors. I'll write out that word. Alpha, and these are adrenergic receptors for both our norepinephrine and epinephrine. So alpha adrenergic receptors. And norepinephrine is the simpler one, so I'm going to do this side of the flow chart first. So norepinephrine binds only pretty much to those alpha adrenergic receptors. You can see that really we have an extreme preference in those beta 2 receptors for epinephrine, so we're not going to think of norepinephrine as binding to them really at all. So when norepinephrine binds to the alpha receptors, the overall effect is vasoconstriction. Okay, so this should have been part of your answer for this warm-up. And this is happening pretty much everywhere, not the brain. And not an overall effect in our skeletal and cardiac muscle, because we're going to see that they're going to be affected on this other side of the diagram. But specifically, we want to remember this is not happening in the brain. Okay, so then epinephrine, we're going to see affect both our alpha receptors and our beta 2 receptors. It's going to make a difference, the concentration of epinephrine. So norepinephrine, we're not worried about concentration. For epinephrine, we are. So we're going to have an effect of lots of epinephrine, and we're also going to have an effect of small amounts of epinephrine. Okay, I'm going to do the small amounts first. So over here on the right, if we have low epinephrine, small epinephrine, we're going to see, if we look on this chart, right, if there's only a little bit of epinephrine circulating around, it's going to be highly preferentially drawn up by those beta-2 receptors. So in low amounts, that epinephrine is going to bind to those beta-2 receptors, 2 adrenergic receptors. And the overall effect here of those beta-2 adrenergic receptors is vasodilation. Okay. And this is specifically happening in skeletal and cardiac muscle. So for small amounts, we're dilating our blood vessels in the skeletal and cardiac muscle. But once we have lots of epinephrine circulating around, we're going to have overflow, right? And it will be drawn up by those alpha receptors as well. Okay, so with large amounts, right, say we have large amounts, we're going to see that we will have epinephrine spill over and bind to those alpha receptors. And the effect of binding to alpha adrenergic receptors, whether we're talking about norepinephrine or epinephrine, is going to be vasoconstriction. So you can directly link in your mind alpha receptor vasoconstriction. Okay. So the receptor type is going to determine our vasoconstriction versus dilation here. Okay. Vasoconstriction. And again, this is happening most places, but not the brain. So this should have been the second part of your answer for this warm-up question. So that mainly vasoconstriction is going to happen as a reaction to norepinephrine binding to alpha receptors. But if you have lots and lots of epinephrine, it will begin to also bind to those alpha receptors, which will also cause vasoconstriction. Questions about this short review? We'll make sure to periodically review this concept as we move forward. Okay, so we're going to move now into the capillaries. Maybe I'll even save this. How do I do that? Great. Okay. So now we're moving into the capillaries. So the capillaries are where this blood is going, part of why we are changing the radius and therefore the resistance of the arterioles. So we're controlling how much blood is going to the capillaries in specific organs and specific regions. So we're going to take a look at capillary anatomy. We're going to look at control of blood flow to and through those capillary beds. And then really crucially, we're going to be looking at the movement of the material across the capillary walls. So this is going to take a bit of brain power when we get to this third point. That's the mathy bit I mentioned. So we'll touch it on this again, basically. That's our other topic. We're going to make sure to review repeatedly, make sure we get some practice with that. Okay. First, we're going to just think about our overarching capillary stuff. Right? So the main role of those capillaries, so we want things diffusing out of the capillaries, out of the blood into the tissues, and we want other things diffusing from the tissues into the capillaries, right? We've talked about this exchange of nutrients into the tissues and wastes out of the tissues and into the blood. Okay. So what we're trying to think about as we think about capillaries is how that happens, like what drives the diffusion, what allows that diffusion to happen. So to understand this, we do have to understand a little bit about the anatomy of the capillaries. So what they look like, how many there are, and specifically what their walls look like is going to make a big difference. So as we said, they're the site of exchange between the blood and the tissue. It's where we're actually moving nutrients in and wastes out. Okay. They're really small. They're 5 to 10 millimeters in diameter. This allows for a small distance for things to diffuse in and out of the tissues, right? Yeah, the substances don't have that physically far to travel, and we'll also see that those walls are quite thin. Another thing we noticed when we looked at the capillaries is that they're quite narrow. We're going to talk about that in a second because we have a caveat there. But first, speaking about the walls of these capillaries, just one cell layer. So one endothelial cell layer, just like a specialized epithelium. We looked at epithelium in lab last semester. They're attached together with a basement membrane, which is basically some extracellular matrix, just sticking them together. This means there's a really small diffusion barrier, as well as having small capillaries, because those substances are only going to have to cross through one layer of cells to get where they're going. That's why it's important that we don't have wrapping of smooth muscle around the capillaries, right? Because then in order to get to the tissue, we'd also need to diffuse through that smooth muscle layer. Okay, so we just have the one endothelial layer here. Okay, so this is our sort of caveat on size. So while they do have a small radius, we're going to see that they have the greatest total cross-sectional area. So individual capillaries are really small, but all the capillaries grouped together in a region, in an organ, or in your body, if you add up their diameters, they become really big. And the reason that's important is that we want to think about the speed of the blood flowing through the capillaries. So we can kind of think of these arterioles carrying blood to the capillaries, and then think about it like a river spilling out into a lake, right? Our individual capillaries are small, but if you add them all together, they're like a really big lake. They have the greatest total cross-sectional area of any of your blood vessels if you added them all together, which means that we have a sort of smaller amount of blood spreading out into this big network, which is part of what allows the blood flow to slow down as it spreads into this big area, this big total network area. And slow blood flow through the capillaries is important because this enhances the exchange. Right? If you had blood rushing past here, rushing past the tissue really fast, you wouldn't have time for the things inside the blood and in the interstitial fluid we'll actually see is what directly abuts the capillaries. You wouldn't have time for them to kind of reach equilibrium in terms of their forces. So it's important that we have this slow blood flow. We have lots of time for things to cross in and out. Okay. So like we said, tons and tons of capillaries. And the total surface area of those capillaries is kind of insane, right? 600 meters squared. Maybe you'd think there's that much space in your body, but I wouldn't, right? So almost every cell is within a millimeter of some capillary. The capillaries are really short as well. They're only about a millimeter long before they drain back into the venous system. Okay. When we're looking at the walls of the capillary, we have this endothelial cell layer, right? Our single cell layer. We're going to see that there are pores between those endothelial cells. And exactly how wide the endothelial cells are spaced, how big those pores are, is going to depend on the type of capillary that we're looking at. We're going to see that some capillaries have smaller pores, smaller spaces, and some have larger pores and larger spaces. So different molecules basically are going to be able to travel between those pores depending what type of capillary we're talking about. We're mostly going to focus on continuous capillaries, at least for now. Okay. So what we're going to see traveling here between our endothelial layers, or between our endothelial cells in that single layer, is protein-free plasma for now, right? It's because proteins are big, they can't fit between cells. Okay. When we refer to all our capillaries together, we refer to them as a capillary bed. So we've seen a chart before of pressure differences across the aorta, arteries, arterioles, capillaries, venules, veins. We saw that that was kind of like falling off a cliff. Here what we're seeing is changes in cross-sectional area. So we can see that this chart is quite different. If we add together all our arteries, they have a pretty small cross-sectional area, but the blood is moving fast through them. They have pretty high pressure, right? Those are kind of inverse properties. We can see that the arterioles rise in cross-sectional area just as they drop in pressure. So that's also kind of the inverse of what we saw in our pressure graph. And we can see that the capillaries particularly have a really large cross-sectional area, just like they had low pressure, right? So it's this sort of first half of this graph that's the inverse of our pressure graph. Now the venules and the veins in size kind of mirror the arteries and arterioles, so that part's not directly related to our pressure graph, but up until we get to the capillaries it is. Okay. We can see that this cross-sectional area is directly the opposite of the velocity of blood through these vessels. So we can see that we have really fast blood through the relatively small aorta, because the aorta is just one blood vessel, right? Quite fast through the arteries, speed is dropping through the arterioles, super low through the capillaries. And then as we start to move blood back to the heart, speeds up again as it moves through the slightly smaller venules and veins. Again, this is adding together all the vessels in the network, not individual venules or individual veins. Okay. So I'm going to draw here. Okay. So I said there are a couple different types of capillaries. So our most common are the continuous capillaries. So that's mostly what we're thinking about. The key to continuous capillaries is that they're going to have small gaps between the endothelial cells. So they're going to allow some small water-soluble molecules to move through, but they're not allowing like proteins to travel through. These are different than our fenestrated capillaries. So we'll see fenestrated capillaries a little bit in the future. The difference with a fenestrated capillary is that we have our cells quite widely spaced. So there are going to be large gaps between our endothelial cells that are basically forming like windows or doors through the cell layer. So I'll pick up my pen real quick. All right, so if I draw my continuous capillary, think of this as a cross section. I'm going to draw my cells now. So if we're thinking about the endothelial cells in a continuous capillary, you can just kind of lay them down like bricks, right? All next to each other. There might be some small pores, right, between the cells, right? But they're very small, not a lot of space. We would continue this all the way around. I get slightly faster, and therefore my drawing will get worse, but I think you get the idea. Okay. This is different from our fenestrated capillary. I'll have a cross section there, because our fenestrated capillary is going to have large gaps between the cells. All right, I'll make these cells slightly larger just so my drawing is faster. But we're going to have these cells much more spaced out, which is why we're going to have room for the proteins, maybe even blood cells, to move between this endothelial cell layer, right? So we imagine protein trying to get out here, right? Protein goes up to this pore, bloop, nope, can't go through, bounces back, right? That space is too small. If we have a protein trying to get out of this fenestrated capillary, it can go straight through this fenestration, which is a window. There are also discontinuous capillaries and sinusoids, which are like kind of an even more extreme version of the fenestrated capillaries. We're not really going to talk about these at all in this course. Okay. So as we're thinking about diffusion across the capillary beds, we want to think about some local control factors first. So this is kind of building on our understanding of what was going on in the arterioles. We are not going to have smooth muscle around the capillaries themselves, but we are going to use smooth muscle around the arterioles and around this sort of initial point of the capillaries. We're going to have something we call a precapillary sphincter that is able to either allow blood flow through that capillary or cut it off. Now this middle bullet point, this meta-arteriole. Meta-arterioles are basically like bypasses that go through the capillary system. They are arterioles, and they're going to act as a shunt. They're going to allow things to kind of skip over the capillary bed. So we'll see what that looks like in a second. Okay, so our meta-arterioles. So I'm going to explain this drawing first. So what we're looking at, let's pull out my pen again. Okay, so here we're looking at blood flow coming from the heart, right? So our heart would be up here, right? Blood flow coming through the heart, branched through the arteries, now it's in the arterioles. So what we see circling around it here, right, are rings of smooth muscle. So we know that the arteriole is surrounded by smooth muscle, which allows us to do vasoconstriction and vasodilation, changing the radius of that arteriole, therefore changing the resistance of the arteriole, therefore changing the flow through that arteriole. So that's one way that we're controlling how much blood gets to the capillary bed, because if we don't have a lot of blood coming through this arteriole, we don't have enough blood to go through the capillary, right? Our returning blood, just so we're clear, right here, we have our venule and eventually our veins will be returning us to the heart. Okay. Here's our capillary bed. So we'll see these narrow capillaries, right? So these are our capillaries. So we see the site of exchange right here, right? So we have things filtering out of the blood at the beginning of our capillaries and filtering into the blood, which we actually call reabsorption at this venous end going into the venule. We'll talk about the pressures that create this kind of system at the end of this packet. Okay, so those are our capillaries. Undo those arrows because they're messy. When we're looking at the control of the capillaries, we're going to see here we have these sphincters, these precapillary sphincters surrounding like the base of the capillary, right? So before we branch into this capillary, we have this precapillary sphincter. Now, if that sphincter contracts, right, we are still going to have blood coming through that arteriole, right? Blood would still like to leave and go back to the heart, right? Because we know there's a pressure gradient there. So what's going to happen if we cut off this capillary bed? So the blood is going to skip it, but it's not going to go travel out to some other organ system. The way it's going to get to that venule and eventually to the vein is it's going to act as a direct connection from the arteriole to the venule. It's going to totally bypass this capillary bed. That's why we're thinking about it as like, I don't know, an overpass, a shunt, a bypass. It lets us skip over this capillary step. Okay. So that's why we have these rings of smooth muscle at strategic locations, because sometimes we aren't going to want to skip a capillary bed, right? Depending on what the tissue surrounding that capillary bed is doing. These rings of smooth muscle, so like these precapillary sphincters, not responding to neurotransmitters like the smooth muscle around the arteriole do. They're only responding to local factors. So we talked about these local factors when we talked about arterioles. Basically, we're thinking O2, CO2, right? So these are reacting to local factors at the precapillary sphincters. So here we have our precapillary sphincters, right? So rings of smooth muscle as we lead into this capillary. They're contracting and relaxing in response to local factors. So if they contract, they constrict the capillary, decrease blood flow to the capillary, which increases blood flow through the meta-arterial. Right? So when we're talking about increases versus decreases blood flow, we want to be careful whether we're thinking about the capillary bed or this meta-arterial, because they're basically going to be the opposite, right? If you're cutting off blood flow to the capillary, that blood has to go somewhere, goes through the meta-arterial. If you relax that sphincter, right, we're going to increase blood flow through the capillary, which would lessen blood flow through the meta-arterial. This is all due to our local metabolites, so O2 and CO2. Now comes the slightly more detailed part. We're thinking about the movement of the material across the capillary walls. And this is really what we're interested in from a physiological perspective. We want to know how our tissues are getting their necessary nutrients and how they're getting rid of their wastes. So that's this exchange function of the capillary walls. That's what they're there for. The most common way we're going to have things moving across the capillary walls is diffusion. So remember, diffusion is just this property of the world, right? Where things that are super concentrated like to spread out, they like to reach equilibrium. So they like to reach equilibrium across membranes. And that's why we see the forces we do when we talk about gradients, like chemical gradients, right? We move down a gradient from an area of high concentration to an area of low concentration. So just due to this property that's true about the world, that stuff likes to spread out. Okay. So we're going to see two types of diffusion in our capillaries. One, we're going to see stuff diffusing directly across the membrane. So remember that our membranes of cells are made of phospholipids, so lipids, nonpolar. We're going to need molecules that are lipophilic, so attracted to fats, so that's going to be like other nonpolar things, are able to diffuse directly across that membrane of the capillary walls, right? So directly across our, right? Directly across our cell, right? So we're thinking not moving through those pores, right? Moving through the cell itself. Things that are lipophobic, right? So lipophobic, we're going to have polar things, right? More water-soluble things. Going to have to go through either channels, so like protein channels in these cells, or potentially, depending on how large they are, through those pores. But so long as we're just going down our gradient, we got diffusion. Okay. Bigger things are going to need other mechanisms of crossing out of the blood into the tissues. The main one we want to think about is something called transcytosis. This is going to happen to molecules we call exchangeable proteins. So exchangeable proteins, ones that we can exchange between the blood and the tissue. So the way transcytosis works is basically like endocytosis. So the cell is going to have to change its shape. That plasma membrane is going to have to invaginate, right? Make a dip, make a little bubble. That bubble pulls off from the membrane, travels through the cell. Then on the opposite side of the cell, we see exocytosis, that protein getting passed along to where it's going. So that's transcytosis, and I think the next slide has a picture. A third thing we see is mediated transport. So that's just really highly controlled transport that happens in the brain. So we barely talked about this in A&P1, but if you remember the phrase blood-brain barrier, the capillaries in the brain are specialized because we really don't want whatever's in your blood getting into the brain. Those concentrations of the extracellular fluid in the brain, that interstitial fluid, is really important, right? We saw how the concentrations of our ions there are determining our action potentials, right? We want to be very careful about what's there. So this is highly controlled, involving our glial cells in the brain. Now we are thinking about the actual pressures that are going to influence the exchange across our capillary walls. Some definitions first. Try not to get these confused in your head. So we're thinking about things moving out of the capillary, so like out of the blood, and into the interstitial space between cells. So when we're thinking about exchange across the capillaries, our like basic diagram is going to look something like, right, we're going to have our capillary. We're going to have, let's just do a big thing like this, right? This is our tissue. Then we're going to have our interstitial fluid. I can type that. All right, so here's our capillary. Here's our tissue. And here's our interstitial fluid. Move that over a little. I'm going to abbreviate that IF. Okay. So this is the situation we're thinking about as we start to think about capillary exchange. So when we have filtration, what we mean by filtration is movement out of the capillary. Oop, different color. Okay, out of the capillary and into this interstitial fluid is filtration. Okay. I'm emphasizing this just because it's important that you think about filtration and absorption from the perspective of the blood rather than like from the perspective of the tissue. If you think about it from the tissue's perspective, you're going to get confused. So this is filtration. We're moving from the blood, from the capillary into that interstitial space. We're moving the opposite direction. Opposite direction. Oop, no, another color. Right, if we're moving from the interstitial fluid into the capillary, we have another word for that. So we're going to call that absorption. Sometimes you may see it referred to as reabsorption. That's going to be a detail we'll get to a little more when we talk about the kidneys. Okay, so filtration, movement out of the capillary into the interstitial space, which is filled with interstitial fluid. Absorption is movement out of that interstitial space into the capillary, into the blood. So the blood is absorbing this stuff. Okay. Now the overall purpose is to distribute our extracellular fluid, right, and to reach equilibrium between the concentrations of all the different metabolites that are in both spaces, in the interstitial fluid and in the capillary. So here we see better drawings of endothelial cells and those capillary walls. So here we see what a capillary would look like if we looked at it kind of from the side, right? We'd see that we have basically tiled endothelial cells. They're pretty close together, but in some places they have little pores, right? So if we then zoom in, and here we're zooming in on like part of this cross section, you can see the pores. We can see that some water-soluble things are getting through, right? Like sodium, potassium, glucose are going through these pores between our cells here. We can see that non-polar things like O2, CO2, and lipophilic things like fatty acids, steroid hormones, remember steroids are also lipids, can diffuse directly across the membrane, right? Lipid-soluble substances are going directly through the cell. They don't need any channels or anything to go through because they can just integrate themselves into those membranes. We can see our large proteins bump into the cells, right? They can't get through. They're too big to go between our cells. So they have to go through via transcytosis. So here we can see the endocytosis portion where they're absorbed by the cell and the exocytosis portion where they are leaving the cell in a vesicle, just the same way they entered, basically. Okay. So that's our capillary wall. So now we're going to think about the forces that are going to create the bulk flow across our capillary wall. So mainly here, we're kind of thinking about fluid, so movement of things like water, right? So we're going to see two main forces. We're going to see the hydrostatic pressure, and we're going to see the osmotic pressure. So the way we want to think about these things, right? I'm going to draw my basic idea of a capillary, right? So we have our capillary, we have our tissue, right? Our tissue, and then we have that space between, that interstitial space filled with interstitial fluid. We're going to have hydrostatic pressure and osmotic pressure, and we're going to be tracking the pressure in both the capillary and in this interstitial fluid, in this interstitial space, right? So to figure out whether we're going to have overall filtration or absorption, we're going to need to compare the forces inside the capillary versus the forces in this interstitial space to figure out whether stuff moves towards the interstitial space or towards the blood. Okay, so each of these spaces, the capillary and the interstitial space, are going to have their own hydrostatic pressure, which is basically the pressure of the fluid pushing on the walls of the capillary, either blood pushing out or interstitial fluid pressing in, right? So we talked about hydrostatic pressure when we were looking at just our basic ideas of pressure and flow, right? The pressure of a hydrostatic column was just those beakers filled with certain amounts of water. So that's the same thing here. Hydrostatic pressure pushing on the capillary walls, either pushing out or pushing in from the interstitial space. Okay. So I'm going to draw those on. When we're talking about hydrostatic pressure, you're going to see it abbreviated P. And then we're either going to have P of the capillary. Oh, I should use the typing part. Pressure of the capillary or pressure of the interstitial fluid. I'll make this small. Sorry, the annotation tool isn't great for subscripts. For the osmotic pressure is going to be the result of non-permeating solutes, so things that can't get across that endothelial cell layer in any way, are going to exert a force on water. And basically, they're going to pull water towards the area where we have a lot of non-permeating solutes. So we talked about this way back when we were talking about osmosis. Okay. But I was drawing on the hydrostatic pressure. Sorry, guys. Okay. So hydrostatic pressure from the capillary. So pressure from the capillary, right? Blood inside the capillary pushes like that. All right. So that's P cap. Okay. And fluid in this interstitial space pushes like that. All right. So that's going to be our pressure from the interstitial fluid, like that. So those are the two hydrostatic pressures. So now we're thinking about the osmotic pressures. So we're thinking about the force exerted on water by things that can't get across our endothelial cell layer. So basically what we're thinking about here are the proteins, those ones that needed to do transcytosis, because we saw the ions sneaking through the pores. We saw lipophilic things being able to cross directly across the membrane. So it's big stuff that wouldn't be able to get through those pores, which are proteins. We'll also refer to them as colloids sometimes in the future. And sometimes we're going to refer to this pressure, therefore, as oncotic pressure. The concept is still the same here, oncotic pressure and osmotic pressure. This is just being a little more specific, that we're talking about the force of the proteins. So that's just kind of a specialized form of talking about osmotic pressure. Okay. I'm going to undo a couple of these circles. I'm going to just keep drawing on here for our osmotic pressures. So I'm going to draw some solutes, right? So basically, right, we have our proteins that are in the blood, right? So if we have a bunch of proteins in the blood, they are going to draw water towards them, right? Because they want to reach equilibrium. They want to be equally spaced out as any proteins in the interstitial fluid. They can't cross the layer, so they need to draw water towards them in order to space themselves out more. Okay, so that's what we're talking about when we're talking about our osmotic pressure of the capillary. So we'll see that these osmotic pressures are labeled as pi cap and pi IF. Okay. So that's what we're talking about when we're talking about pi cap. Proteins dissolved inside our blood. And the reverse happens from the interstitial fluid as well, right? So if we have a bunch of proteins, pick a different color so that looks a little more different. Okay, we got some pink. Okay, so if we have a bunch of proteins dissolved here in the interstitial fluid, they are also going to pull water towards them, right? So this pull of these proteins dissolved in this interstitial fluid is the osmotic pressure of our interstitial fluid. So that's pi IF. Okay. So in order to figure out, as we will later, whether overall we're going to see movement of fluid, of water into the interstitial fluid or into the blood, we're going to have to compare basically these four variables: the hydrostatic pressure of the capillary and of the interstitial fluid, the osmotic pressure of the proteins in the capillary versus the proteins in the interstitial fluid, because each of these is either pushing or pulling on water, right? And if we're thinking about a single water molecule, it can only move in one direction, even if there are four forces acting on it. So we're going to have to consult all of these to figure out overall where our water is moving. Okay. I think I intended to draw here on this slide, but we drew it on the previous slide with the words, so that's okay. Okay. So thinking about the hydrostatic pressure, we actually know some specific numbers that basically remain the same when we're thinking about capillaries. Okay. So here we have our values. So we're going to start actually thinking about some of the math, right? So when we have our capillary, on the end, attached to the arteriole, so forget about meta-arterioles for a second. We're just going to do a little diagram where we have an arteriole on one end, and we're going to have our vein on the other end. Our venule is here. Okay. I'll make this part blue. Okay. So there's our capillary. All right. We're going to see that there's higher pressure at the end attached to the arteriole. So the blood is rushing in here at the arterial end. And at the venous end, we're going to see a little less pressure. And specifically, usually we have about 38 as our pressure at the arterial end, but at the venous end, we have about 16. So we can see that there's a pressure drop across this capillary. Whenever we're thinking about capillary hydrostatic pressure, these are our values for filtration, right? So we have, when we're eventually going to add this into our equation, we're going to have 38 millimeters of mercury as pressure out, so favoring filtration here, versus 16 millimeters of mercury favoring filtration here, right? So both of these are pressures pushing out, although our net movement of water is going to be different on the arteriolar end versus on the venous end. So the capillary hydrostatic pressure is always a pressure favoring filtration, favoring movement out of the capillary, regardless of what the total end picture is going to look like. Now, we're also thinking, we're also thinking about our interstitial fluid, right? So here's our tissue. Oh gosh. Okay. And here is our interstitial fluid, this space between. Okay. So the interstitial fluid here also has a hydrostatic pressure. So that's like how much fluid we have here. Its pressure is not going to change much from the arteriolar end of the capillary to the venous end of the capillary. It's just this continuous space with itself. It's bigger. It's mostly in equilibrium across the end of the capillary. So it's going to be between 0 and 1 milligrams of mercury across this length. So say we got, let's pick 1, right? So 1 here, but it's still 1 on the other end here. So, but both of these are pressures for reabsorption or absorption. We're going to think of those as equivalent for right now, although they won't be when we get to the kidneys. Okay. So those are pressures moving us towards the blood. Okay. So the interstitial hydrostatic pressure is always pressure for reabsorption. Okay. So we're going to stop here and we'll pick up on Wednesday talking about a little bit more about the osmotic pressure in the capillary and in the interstitial fluid, and we'll do a little bit of a practice problem as well to make sure we got this down. Okay. Oh, and just an update about the quiz. You should now have a view now that the quiz is closed. If you click back into it, you should see your answers and see the correct answers so that you're able to study from that. So hopefully that works for everyone. If not, we will brainstorm.